Good morning, everyone. This morning I'm going to tell you about global neuroimaging studies with diffusion MRI, tractometry, and generative AI. And this talk was also part of the DiPy medical imaging workshop. And so if you're interested, the link is there at the bottom. It's a wonderful workshop that gives you a guided tour of all the tools available for analyzing brain microstructure using DiPy. A really exciting course. It lasts 5 days. So today's talk is going to be on global neuroimaging studies with diffusion MRI, tractometry, and generative AI. Even though I'm presenting it, there'll be a lot of work that has been led by others, particularly Dr. Bram Chandio, Dr. Julio Villalon, Wendy Feng, Dr. Leila Labulsi, Dr. Connor Owens-Walton, and Gaon Kim in our lab. And there'll also be work presented from the Jahanshad lab at USC, notably by Iyad Baghari, Alyssa Zhu, and Talia Nir. And this has also all been supported by the Indiana University DiPy team, notably Eleftherios Garifalidis and Jarek Haraslag. So for the next 45 minutes, I'll tell you a little bit about a number of international projects that study brain microstructure. We'll introduce, first of all, what is ENIGMA. It's a giant neuroimaging consortium studying over 30 brain diseases. We'll talk about how you can use diffusion MRI to study brain diseases with a lot of examples from ENIGMA. We'll also talk about how brain microstructural metrics vary throughout life. So this will involve a normative study of diffusion MRI in 50,000 subjects led by Julio Villalon. And then we'll talk about some technical issues, such as how the scanning protocol affects diffusion MRI metrics and how to compensate for that using harmonization methods. One such method is eHarmonize, developed by Alyssa Zhu and Neda Jahanshad. Also some applications of bundle analytics. So as you look into diffusion MRI with tractometry and reconstruct the fiber bundles of the brain, there's a number of techniques that have highlighted brain differences in Alzheimer's, Parkinson's, bipolar disorder, and autism, and we'll review some of the findings there. Last of all, we're going to talk about generative AI, which is a very powerful approach that can be combined with tractometry for anomaly detection in the brain and even for quality control in your analyses. So what is ENIGMA? It's a massive international study of the brain that's been going on for a little over 15 years now. Since 2009, ENIGMA has published the largest neuroimaging studies of 15 brain disorders. The overall goal is to find factors that are helpful or harmful to the brain by analyzing brain scans and genetic data from now over 100,000 people in 45 countries. And a little over 2,000 scientists all collectively working on this in 51 different working groups. And this has led to the largest neuroimaging studies of epilepsy, Parkinson's disease, ataxia, and traumatic brain injury. And in psychiatry, bipolar disorder, major depression, schizophrenia, PTSD, and substance use disorders. And in the neurodevelopmental spectrum, obsessive compulsive disorder, ADHD, autism spectrum disorder, and some neurogenetic conditions such as 22q11 deletion syndrome and some infectious diseases such as HIV. So ENIGMA's analyses of 100,000 brain scans has two main goals. The first is to understand how now 33 major disorders affect the brain. Now, today's talk, I'm going to concentrate on diffusion tensor imaging, or DTI, but there are also ongoing analyses using standard MRI, anatomical MRI, resting state functional MRI, EEG and MEG data, but we'll talk about those another day. Another thing that ENIGMA does is try to understand how genetic variation affects the brain by screening millions of markers across the genome using various genetic analysis methods such as genome-wide association studies or epigenome-wide association studies, or even looking at copy number variants in the genome to find genetic markers and environmental factors that affect the brain's development and aging, your disease risk and treatment response. So not every study needs to be done using ENIGMA's framework. This very large-scale collaboration is particularly suitable if you are asking a question that requires very, very high statistical power, such as cracking the brain's genetic code, looking at the genetic markers that influence the brain that have been impossible to solve before. It's also very efficient. The studies use massive infrastructure here and overseas in 45 countries, which is very useful if you're doing deep learning or cooperative machine learning. And also, it's not just the data. I mean, the collective intelligence of all the aggregated scientists who are working on this is really very, very highly significant. So this is where the data is. People are scanning the brain with MRI all over the world. and the scans are accumulating in the locations that you see here. The color key shows the main diseases that these or disorders that these sites study. There is an incredible range of data that's spread across the world, covering a large number of different diseases. And we'll tell you a little bit about some of the clinical topics that ENIGMA focuses on. So in the middle, you see neurological disorders such as ataxia, epilepsy, Parkinson's disease. Around the perimeter, you will see some psychiatric conditions such as schizophrenia, bipolar disorder, and major depression. Over there on the right, if you want to study panic disorder and social anxiety with neuroimaging, you can. And then on the bottom, you'll see some developmental or primarily neurodevelopmental disorders such as obsessive compulsive disorder and autism. And there are also very large-scale studies with neuroimaging in ENIGMA of these. Now, not all of these conditions are homogeneous. Brain injury, for example, can occur in the military or in sports or in children. So there are very large numbers of subgroups dedicated to looking at these questions in particular. And these are all aided by technical groups. So a lot of the neuroimaging methods, diffusion MRI, which we'll focus on today, but also resting state functional MRI, require specialized techniques to process the scans. And even groups that focus on the genomic data on the right will help people across the world to analyze these data and relate them to the brain. So here's a sort of map of the groups in ENIGMA. The newest ones focus on chronic pain, including migraine, interventional techniques such as neuromodulation to ameliorate the brain disorders, contemplative medicine, including meditation as a way of alleviating some of the symptoms in psychiatric conditions such as depression and chronic pain. And then also, as I have already mentioned, ENIGMA Environment, which examines the effects of pollution and other environmental factors on brain health. Here are some reviews of ENIGMA, focusing on the logistics or the genetic projects or even some of the neuroscience findings, which you're very welcome to check out. Many papers per year, about 70 in fact, including this collated special issue on ENIGMA in 2022. And also some popular media have covered this as a collaborative approach to study the brain, essentially by crowdsourcing or allowing the participation of people across the world in data analysis. And here are some of the folks leading the ENIGMA analyses. Some of them you may recognize. So let's start with an overview of an ENIGMA study. And this is a picture of the areas of the brain where cortical thickness, the thickness of the cortical gray matter, is reduced or lower in a range of different brain disorders. So you can see major depression, bipolar disorder, schizophrenia, alcohol use, obsessive compulsive disorder, ADHD, autism spectrum disorder, epilepsy, and the neurogenetic condition 20q11.2 deletion syndrome. The colors show the amount of difference on average in people with the condition versus healthy controls. Red colors denoting lower gray matter thickness on average, blue colors higher gray matter thickness on average. And these are averages drawn from data sets throughout the world. You can also buy a t-shirt and if you really like the data and you want to look at it a lot. And just to sort of say how these studies are coordinated, you may be a scientist or a lab shown here as a dot, and you may collaborate with other scientists analyzing either the same type of data, such as DTI, or with scientists analyzing a different type of data, such as functional MRI, or even a different disease. And this sort of modular hierarchical network facilitates the exchange of information on data processing, cross-modality and cross-disease comparisons. So many methods are used across ENIGMA. Today's talk will focus on diffusion MRI, but there are pipelines and workflows for analyzing functional MRI, MEG, EEG, voxel-based analyses of brain data such as CAT12 and VBM, and analyses of the cerebellum and the sulcal pattern of the cortex. And so if you want to check out some of the protocols, they're all linked here. So what do ENIGMA members do with their scans? Well, if you have an anatomical MRI data set or a diffusion MRI data set, you can use a workflow to split the brain up into regions and measure various aspects of interest, such as the thickness or surface area of certain cortical regions, the volumes of parts of the subcortical anatomy, such as the hippocampus. Or with diffusion MRI, you might be interested in microstructural measures, such as fractional anisotropy or mean diffusivity, either for particular regions of interest, in which case you'd use TBSS, or tract-based spatial statistics, which is implemented in the FSL toolkit. Or you could look at these with tractometry, which we'll talk about later on. Once you've measured these parts of the brain, you might be interested in testing whether they're associated with clinical measures or risk factors for disease or possible modulators of disease, such as treatment. So each site participating in ENIGMA runs a multiple regression statistical analysis. They're testing associations between the brain measures and the disease measures, also adjusting for age and sex. And when each site has finished this, the coordinating analyst runs a meta-analysis where the effects are combined across sites, and each site's contribution depends on its sample size, and this makes sure the effects are reproducible and can boost power to pick up effects that individually the sites might struggle to identify. So let's do a quick survey, a guided tour of brain differences in different neurological and psychiatric conditions, as seen with, first of all, anatomical MRI, which measures things like cortical thickness and hippocampal volume, and then diffusion MRI, which, as I've mentioned, examines the white matter microstructure. It may be parts of the brain, such as the genu of the corpus callosum or the cingulate hippocampal bundle. And the measures, which I'll also explain, might be things like mean diffusivity or the fractional anisotropy of diffusion, which we'll talk about next. So here are some of the studies that we'll review. We've talked about these different disorders. Let's look at some of the results in the brain. So here's the ENIGMA schizophrenia group. It's led by Jess Turner and Theo Van Erp. 33 sites worldwide have been collecting anatomical and diffusion MRI, a huge number of patients. Let's say the sites measure, first of all, the thickness of the cortical gray matter in different parts of the brain using FreeSurfer, a very widely used toolkit. And let's say they want to ask the question, do people with schizophrenia on average tend to have lower or higher or different cortical thickness in these regions? Well, they do. So in a study of over 8,000 people, it was found that the majority of the brain shows thinner gray matter in schizophrenia. The effect size can be described using Cohen's d, which is a measure of how much the group difference is. Cohen's d of minus 0.5 shows that the schizophrenia patient group deviates from healthy controls by about half a standard deviation. They're about half a standard deviation lower than the controls. A Cohen's d of 1 would indicate that the two groups differ by 1 standard deviation. A d of 2 would be 2 standard deviations, and so on. So standard deviations are equivalent to z-scores, and this shows the magnitude of the group difference. If you look at the sites contributing to the group, and there are 33 of them, all but one show thinning of gray matter in schizophrenia. And then at the bottom there, when all this data is meta-analyzed, you get an aggregate measure of the effect, a Cohen's d of minus 0.55. So let's look at the different disorders. First of all, looking at the structure of the brain. So in major depression, there are mild gray matter deficits in the limbic system and the frontal cortex. In bipolar disorder, there's a much more severe pattern of cortical thickness reduction, including in the frontal cortex and many other areas. In schizophrenia, very, very severe, significant gray matter loss over the majority of the brain. In neurogenetic disorders, including this one that put you at risk for schizophrenia, the patterns can be very different. And so blue denotes higher gray matter in many of these different regions. Epilepsy, a neurological disorder associated with seizures, there's widespread gray matter reduction. Obsessive compulsive disorder, some frontal deficits. Autism, actually excess gray matter in parts of the frontal cortex and the temporal lobes. Substance use varies in its effect on the brain. Alcohol use in particular is associated with thinner frontal gray matter. And then if you look at the whole mosaic of different disorders, you'll see how different they are. The patterns of cortical anomalies are certainly very distinct and different for the different brain disorders. You can compare these and look at the signature of volumetric difference in these 4 disorders. Notice how they differ in the degree of differences and also the anatomical profile. There's a distinct caudate putamen effect in ADHD, which is not seen in the other disorders. And this comes from combining data, actually from over 20,000 people in 33 countries. So now let's move on to diffusion imaging. So for those of you who are not familiar with it, diffusion imaging is a type of MRI that's sensitive to the microstructure of the brain. It's often used to study the white matter or the connections of the brain, and reveals properties of the white matter that would not be evident using standard anatomical MRI. When we analyze data from multiple sites in ENIGMA, we use the ENIGMA-DTI protocol. This looks at various regions of the brain, and in each of them examines different metrics or measures of brain microstructure. Some of these are computed with a DiPy toolkit, others with the FSL toolbox, and they include diffusion tensor imaging measures, FAMD, RD, AD, diffusion kurtosis measures, MAP, MRI, and NODI, and we'll talk a little bit about what they are next. So diffusion-weighted MRI measures the 3D diffusion of water molecules in the brain. And in an unrestricted medium, such as a glass of water, the diffusion of water is isotropic. It's the same in all directions. This is known as isotropic diffusion. And if you were able to measure it, you can imagine that after a certain amount of time, a water molecule in the center of the field of view might, through Brownian motion, have diffused a certain distance. And the probability density of those diffusion distances is mapped here using a spherical Gaussian distribution for a certain amount of time. Now, if you're looking at a tissue, such as neural pathways in the brain, the water diffusion is constrained. And so it's more likely that water will diffuse along the axons of the brain's neural pathways. If these pathways are heavily myelinated or insulated, there's very little diffusion of water transverse to the axon. And so you'll see that when a neural pathway is present or an axon is present, it causes a sort of anisotropy or differential diffusion in different directions. And this can be measured using diffusion imaging indices such as fractional anisotropy or FA. Also, when there are multiple fibers present that are crossing, you can see the influence in this figure borrowed from Patrick Hagman on the bottom left, where the diffusion profile is influenced and is directionally constrained along different directions. Now, we're interested in this because brain diseases also affect these measures in interesting ways, so we can use them to detect disease. We also have to understand how these measures depend on the tissue type, and in fact, the imaging protocol and the mathematical model used to reconstruct diffusion. So we'll talk about some simple models of that first of all. So when you're scanned with diffusion MRI, the diffusion-weighted signal is very complicated. It can be reconstructed using the statistical-Tanner equation or Peter Basser's variant of that, which incorporates the diffusion tensor, or even more advanced directional diffusion models to reconstruct the three-dimensional orientation density function, which encodes the probability of water diffusion in different spherical directions. And this can then be used in conjunction with methods called tractography to reconstruct or follow the paths of neural pathways in the brain. Now, there's an entire course on this, so I'd very much encourage you to check out the DiPy course on the internet. It's been very widely acclaimed by all of my trainees who attended it, and it discusses all of the incredible range of algorithms that are used to look at brain connectivity, the microstructure of these connections, and even their geometry. This is work from the Center for Magnetic Resonance Research in Minnesota, led by Camille Urbil, with tracks reconstructed by David Shattuck at UCLA. So what does water diffusion or apparent diffusivity in the brain look like? Well, if you were to take a coronal section of the brain that goes through the ventricles are filled with fluid. So you'll see in the middle, there are areas that water diffusivity is very high because they're essentially areas of fluid. But then in areas of the brain that have brain tissue, the water's diffusion is not only very constrained, but it's also aligned along certain directions. So the colors here denote the direction, with red colors denoting left to right in this instance, green colors denoting more anterior posterior, and blue colors denoting diffusion that's primarily in and out of the plane that we're looking at. The diffusion tensor is actually a very simple model of apparent diffusion in the brain. There are many higher order models that you can fit. So one of them that models the orientation distribution function, you can fit a mathematical series to the diffusion profile at each point, using, for example, spherical harmonics, which are a complete orthonormal basis on the sphere. And you can see just incredibly detailed patterns of diffusion in different brain regions. The very heavily myelinated corpus callosum is up there at the top in red, and you can see it's one of the major fiber tracts in the brain. And obviously, the properties of diffusion can be quantified in these different regions. Anyway, how do we measure diffusion and turn it into useful mathematical indices that might possibly reflect disease or aging or development? So the simplest model of water apparent diffusivity in the brain fits an ellipsoid to it, a little bit like a football. Mathematically, the parameters of a three-dimensional Gaussian distribution are estimated that are fitted to the diffusivities along different spherical angles. A diffusion tensor is represented mathematically by 9 coefficients. Some of them are redundant, it's symmetric, so there's actually only 6. And then using eigenvalue decomposition of this tensor, because it's symmetric, you can derive some measures that essentially quantify what is going on with the diffusion. So the eigenvalues of this diffusion tensor, lambda 1 to 3, feature in some formulae. And the formulae there on the top right can be used to compute fractional anisotropy, which is a measure of the directionality of the diffusion. Axial diffusion, which is diffusion along the major principal axis of diffusion. Radial diffusivity, which is the diffusion across the axonal tracts, which of course will be reduced when they're myelinated. And then mean diffusivity, which is an average of all the diffusion tensor eigenvalues, and is actually a pretty good reflection of fiber microstructure, which often goes up in disease. So when the cells deteriorate, the microstructural barriers to diffusion tend to be broken down. And then there's much more diffusion. So mean diffusivity, or MD, goes up. And if you want to look at these visually, the bottom right shows, first of all, a T1 weighted standard anatomical MRI. The map next to it, FA, is a picture of fractional anisotropy. I mean, that doesn't look quite as clear as the anatomical image, but it's the measures that you see are the value of the FA coefficient at each point. Look at how high it is in the corpus callosum. So the bright spots there, where the FA is highest, it's around about 0.6. On the bottom, you'll see axial, radial, and mean diffusivity maps. They all look a little bit different. And the map on the top right is a color map. As we mentioned, if the diffusion is primarily in the left-right direction, it's red. If it's primarily in the anterior-posterior direction, it's green. And if it's primarily in the superior-inferior direction, it is blue. So if you're not familiar with DTI or diffusion tensor imaging, I'd strongly recommend you read a really nice paper by a former colleague of mine, Jeffrey Alger at UCLA, in the Journal of Neuroscience in 2012, and the link is there. This article summarizes DTI methods in terms that can be immediately understood by the neuroscientist who has little previous exposure to DTI. And it's great. I borrowed this figure from that review. So if this is a little bit more than you're used to looking at, that's a very nice reference to go and have a look and get you up to speed. Okay, so in ENIGMA, there's an ENIGMA-DTI protocol. This was based on the Tract-based spatial statistics toolbox by Steve Smith and the FSL group in Oxford. It was modified by Neda Jahanshad and Peter Kochanov and others over the years to be useful for large-scale studies in ENIGMA. It uses an ENIGMA-DTI template, and it also measures diffusion MRI quantities in certain regions of interest in the brain. So if you look at the bottom there, there's an atlas, the Johns Hopkins University JHU Anatomical Atlas. And you can ask the question about whether these metrics of diffusion are different in various diseases in each of these sectors of the brain. And we use the standard TBSS process where measures are quantified on the so-called skeleton, which is a sort of eroded version of the white matter. And this is done for FA and the diffusivity measures. There's also some quality control. But of course, the interest at each site is to see Are there any associations between these white matter metrics and variables of clinical interest? It might be a disease or a symptom or a treatment effect. And you would use inverse variance weighted meta-analysis to combine the effects across sites. So if many sites are participating in the study, you want to weight the information from each, usually according to their sample size, and then you'll get an aggregate conclusion. So a vast range of DTI studies have been completed by ENIGMA groups working on all these disorders that you're seeing here. Many sites participating, different disorders being analyzed. So we'll do a little bit of a survey of the findings of these different studies. And if you're interested in the primary references, here they are. There are 11 papers published on diffusion microstructure in different brain diseases, and the links can be clicked to see what's happening in each disorder. Okay, so let's look at the ENIGMA Schizophrenia DTI study. This was a study that was led by Sinead Kelly and Gary Donahoe, the National University of Ireland in Galway and at USC. It had 107 co-authors and data from 14 countries. And we'll have a look at what's going on in schizophrenia in a little over 4,000 scans from across the world. So, as we mentioned, the FA, which measures the fractional anisotropy of water diffusion, is usually high in very heavily myelinated neuronal pathways, such as the anterior corpus callosum. You'll see that in schizophrenia, the FA, which is a measure of the directionality of diffusion, is lower everywhere. Everywhere in the brain, with a couple of exceptions, you'll find that the effect sizes for the effect of schizophrenia are as high as -0.4, which is about half a standard deviation below normal. Very similar effect in terms of its degree and severity to the cortical thickness differences that we described earlier. The effects in the frontal regions are strongest, which fits with the hyperfrontality hypothesis of schizophrenia. But as you'll see, very strong effects, no difficulty at all in finding these. And just looking at the distribution of effects across sites. So each site here completed the workflow, and you'll see that every single one of them, perhaps with one exception, found that the FA was lower in schizophrenia patients than controls. This is a forest plot. It really shows the effects at every site. They really do agree. And this was a paper published in 2017, again by Sinead Kelly, in a little over 4,000 people. Now, you could say, well, what about the other diffusion metrics? FA is lower, so those blue bars there show how much it's lower in the brain's white matter overall, and in various regions of interest, such as the body of the corpus callosum as a whole, the anterior corona radiata, and the genu of the corpus callosum. So they're very convincing differences. The other measures, such as mean and radial diffusivity, are also abnormal. They're abnormally high rather than abnormally low, which generally happens in disease. As we said, the barriers to diffusion tend to have either broken down or be developmentally insufficient. So remember that for these diffusion measures, often the direction of effect in disease is opposite to each other. Radial diffusivity is somewhat redundant with FA. The measures correlated in this study at about 0.83. MD is a little bit less correlated, but also elevated in disease. And so this is giving you a range of metrics of disease that are somewhat complementary, but also give you some independent information as well. Now, one of the questions that reviewers often ask is, the effects that you find in disease depend on the size of the region of interest. I mean, do bigger regions of interest show bigger differences in disease? Does it depend on the scanning protocol? I mean, maybe a better scanner is more effective at detecting the disease. And is it always consistently found across all the sites in the study? So just to go through these in turn, there's very weak evidence of greater effects in the larger regions of interest in the brain. Very tiny coefficient, R squared of 0.15. There's also not a huge effect of protocol. In a high-end protocol such as that used by the Human Connectome Project, you do find a very slightly higher effect size for the disease with these higher signal-to-noise protocols. But very generally, I mean, these are measures like mean diffusivity, which are quite admirably estimated with a low-end protocol. So you'll find that they're rather consistent. In fact, all of the studies on the right, you'll see the effects, even though they're using different field strength scanners and different voxel sizes. And so for the most part, they agree. Now, data from one site in this study was hugely off, but you can see in the forest plot that's easy to identify. And they found that they'd miscoded the data and then later fixed it, and then everything was in a consistent direction. How do measures from DTI compare in terms of the degree of difference in patients versus controls? As you can see on the left, the winning DTI metrics, the ones that showed the greatest difference between schizophrenia patients and controls, were the fractional anisotropy in the anterior corona radiata, which is a very large white matter pathway, and the body of the corpus callosum. You'll see that on the right, similar measures of difference are shown for the hippocampus, volumes of other parts of the brain. There's something interesting here. Medications do not seem to affect the DTI measures, at least in our studies so far. So you can be sure it's a disease effect rather than a medication effect. Whereas with standard anatomical measures, such as the size of the basal ganglia components, the caudate, putamen, and pallidum, we often find that the medications used in psychiatry to treat the disorders, such as second-generation antipsychotics, olanzapine, for example, they do tend to boost the volumes of these parts of the brain. So it's sort of an interesting difference between DTI and standard anatomical MRI, that the medications do not seem to strongly affect the DTI. How do different diseases differ? So if you compare schizophrenia with major depressive disorder, MDD, you'll find that the effects on the brain in schizophrenia are rather a lot worse. In orange on the left, you'll see the effects of schizophrenia. In blue on the left, you'll see the effects of major depression. They're still quite significant in major depression. This is a study, again, by Sinead Kelly and Leanne Schmal and the ENIGMA Major Depression group. There's fairly sizable differences in major depression. If you took a condition such as the neurogenetic disorder, 22q11.2 deletion syndrome, this is a condition that puts you at risk for schizophrenia. And notice how the profile of differences in different parts of the brain have a very different anatomical profile. I mean, they're not particularly alike. And so again, it speaks for a sort of unique pattern of biomarkers for each disorder that we've been studying. Now, this is a very interesting study led by Ryota Hashimoto and Daisuke Koshiyama in Japan. So they lead the Kokoro Consortium, and it's a study across Japan, many centers, as you see on the top right, where they were studying the same disorders, 4 major psychiatric disorders, with, in this case, DTI, in a little under 3,000 people. So how do you think the results compared between their study and the ENIGMA study? Again, the people in each study don't overlap. Well, here it is. And so this is Ryota Hashimoto at a conference in Fukuoka in Japan, showing that the DTI differences in schizophrenia in Japan follow essentially the same pattern as had been discovered in the primarily North American, European, and Asian cohorts. So just to look at this in some detail, the regions of interest of the brain, which are shown according to the key, the effects of schizophrenia on each of these is shown in ENIGMA on the left and Kokoro on the right. Now, Kokoro is a slightly smaller study, so the error bars or confidence intervals on the effects are slightly broader. But you would have to admit that pretty much all of the regions of interest shown in orange follow approximately the same pattern of deficits in ENIGMA and in Kokoro. This study shows that this is the Kokoro sample again, the effects of schizophrenia on microstructure were greater than the effects of bipolar disorder, which in turn were greater than the effects of depression. In all cases, mean diffusivity was elevated abnormally. FA was lower. And you'll see in their forest plot from the sites in Japan, they estimate an effect of schizophrenia of about negative 0.44, which is actually rather similar to the effects that were found in ENIGMA. Now let's look at some other disorders. So Parkinson's disease is a primarily motor disorder that can occur late in life as a result of a neurodegenerative process. So you might expect, and this is a study of diffusion MRI data from 17 cohorts led by Conor Owens-Walton here at USC and his colleagues. You might expect that the same pattern of deficits, low FA, high MD, would be found in Parkinson's disease. And in fact, it is. In the late stages of Parkinson's disease, that is exactly what is found. If you use a Parkinson's disease staging system called the Hoehn-Yahr scale, in which stage 1 is comparatively mild Parkinson's disease, all the way up to stages 4 and 5, which are very severe, you'll see that in patients at different stages of the disease, they have progressively greater abnormalities going towards lower FA and higher MD. But you'll see this rather curious effect at the top, that patients with the earliest stage of the disease seem to have an opposite pattern of white matter microstructure, which could either be an inflammatory effect or an early protective effect or even a compensatory effect. This is still unknown as to what precisely is going on. We're very interested in understanding it, why there would be a higher than normal FA in the earliest stages of Parkinson's disease. This study of people living with HIV, the human immunodeficiency virus, examined whether or not people with HIV, even when they're medicated with antiretroviral treatments, whether they exhibit any microstructural difference in the brain compared to typical matched controls. Talia Neer and colleagues examined DTI across 10 neuro-HIV studies across the US, South Africa, Australia, and even in Serbia, and asked the question about the pattern of brain deficits and whether it links with interesting serological parameters such as T cell count or viral load in HIV. And it does. So this is very much more typical pattern that we've seen in the other disorders as well, of lower FA, higher MD and RD. Interestingly, affects pretty much all regions, which you would think might be the case in an infectious disease. The degree of difference is associated with CD4 T cell counts, but not with viral load. And there's also evidence for faster brain aging in people who are HIV positive. And you'll notice that there's a superscript here, FA-TDF. So they decided not to look at the standard measure fractional isotropy. They looked at one that's a little bit better. I'll tell you a little bit about what is going on with that. So as we mentioned before, the diffusion of water in the brain can be modeled simply using a three-dimensional Gaussian distribution there on the left, either uniform in all directions or directionally constrained. And the use of a diffusion tensor, a very simple model of diffusion, enables you to compute the tensor eigenvalues. There, the lambdas on the bottom left. And using Peter Bass's formulation, which is now 30 years old, you can derive measures of directional constraint in diffusion, such as FA. Now, the problem is many voxels contain multiple fiber directions. There may be fibers that are crossing at right angles. And so in reality, there's a mixture of diffusion components. And the tensor distribution function, which was developed by Alex Liao in magnetic resonance medicine 2009. And since then, there have been similar models by Baba Vimuri and others that have modeled diffusion as a continuous mixture of densities. This fits a probabilistic ensemble of tensors that optimally explain the diffusion data. And you can imagine weighting the FA or the anisotropy of diffusion in proportion to the fractional contribution of each of these tensors. And so there you are on the bottom right with a formula. The TDF-based estimate of FA integrates over spherical angle the tensor orientation density function as a function of the spherical angle theta, multiplied by the FA of that tensor. And that spherical convolution gives you a corrected measure of FA, which we call FA-TDF. So let's look at it. On the left there is the usual tensor estimate of FA. The TDF measure of FA is much brighter. You will see that in areas with fiber crossings in particular, it picks up the signal a little bit better than the standard model of FA based on diffusion tensor imaging. And actually, the associations with disease are also much easier to detect. So let's look at an example. This is a study by Talia Neer of Alzheimer's disease, in which the FA based on the tensor distribution function, the FA-TDF, shows much stronger associations. The maps show differences between patients with Alzheimer's disease and normal healthy controls, associations with clinical dementia rating, and even associations with hippocampal volume. And you'll see that the adjusted measure, so to speak, has much stronger and more significant associations in each of those cases. Okay, so let's look at some other disorders. This is a study of autism spectrum disorder by Gowan Kim and Kathy Lawrence at USC. They ran the ENIGMA-DTI pipeline to look at some of the standard metrics. They all go in the expected direction. In autism, there's a widespread reduction in FA. In other words, it's higher in neurotypical individuals than people with autism. And you can see some of the results there. Now, one of the things that I've glossed over is that some of these sites don't find the same results for associations between disease. And a prominent example is the ENIGMA OCD study. So Fabrizio and Federica Piras and Gianfranco Spalletta in Italy ran a study where they found that the microstructural differences in obsessive compulsive disorder depend on the age of onset, the duration of illness, and also medication. And so if each of the studies had been published individually, the results would seem to be inconsistent. This has also been termed by Ioannidis and others as the reproducibility crisis. And it often means that really the effects of disease depend to some degree on the context and other factors to do with the disease course that might also be interesting to model. Now, if this is the case, we want to be able to understand how these parameters affect the diffusion measures as well. So this led to a bit of a sea change in ENIGMA towards something called mega-analysis. So what is that? So if you want to move beyond meta-analysis, a mega-analysis centralizes the individual data from all of the site scans, or anonymized individual measures are all sent to you if you're the analyst. And this allows much better modeling of the effects of site and scanner and scanning protocol on the brain measures. You can also apply some very advanced deep learning measures to the scans, such as generative adversarial methods, latent diffusion models, to harmonize images from different scanners. But most of all, it lets you study effects of medication, treatment, interventions with much more statistical power. So let's look at a review of this. So Andrei Zugman, who co-leads the Generalized Anxiety Disorder Working Group for ENIGMA, wrote a review of the ways in which you can analyze multi-site data. It's very interesting. So this covers mega-analysis and explains how that is actually done. So when data are shared in a multi-site analysis, there's sort of 4 levels of data sharing that could be followed. In the first 2, you essentially only have the effect sizes at each site. So in a literature meta-analysis, you might be looking up the effect sizes of published studies. There's no coordinated analysis at all. The second degree of coordination might be that everyone uses the same analysis protocol, but only the statistics of effect from each site are sent to the lead analyst. The third level of coordination might be that people can send you individual measures they derive from the raw images, and so that's being done more and more in ENIGMA. Or people can actually send you the raw images, and with more data sharing, better modeling can be done. And we'll have a look at one example of that better modeling next. So if you centralize multi-site data, and we're talking about the measures of the brain from different anatomical regions, you can harmonize data coming from different sites. So this is a study led by Sean Hatton for ENIGMA Epilepsy, where he harmonized DTI data from 24 different sites to correct for scanner-specific variations in fraction isotropy and mean diffusivity. Now, why does this happen? So incoming data from each site is not drawn from the same statistical distribution. So there's often some methods that are used, one of them called ComBat, to adjust the data histograms, or more precisely the residuals of a statistical model, across sites before pooling the data. So let's have a quick look at what is going on. So on the top left, there's the FA measures from different regions of interest And the colors are different sites. So some of the sites in the study tend to have higher FA. They're not entirely different, but there's a shift as well as a sort of scale of the histogram. And it'd be preferable if we could sort of align this data so that the measures were comparable from one site to another. So if you look on the top right there, we've applied to each histogram a so-called scale and shift operation where the variance is scaled to match the common reference, and also the mean of the distribution is shifted to align it with the common reference. And this can be done for each measure to make sure that the statistical distributions are more in line with each other. Now, one analysis toolkit to do this is called ComBat. There's a lot of variations of this. It was originally applied to microarray gene expression data to correct for batch effects, then applied to DTI data. Fortin et al., a very seminal paper there that applied this to harmonized multi-site DTI. And there's various incarnations of this, ComBat-GAM, CoVBAT, numerous of these. Depending on the situation you're in, you can use them to remove scanner effects on your data. So if you're a visual person, this will help you understand what is going on. So on the top left, let's say you're looking at the effect of age on a DTI measure. And let's say in general, the measure declines with age, and the colors are data from different sites, the spots are data from different individuals within those sites. You'll see that on the top left, there's a pretty clear difference in the data coming from the site that is in blue, and the site that's in red, and the site that is in yellow. But we believe that the underlying biology of aging is somewhat similar across those sites. The reason is that there's just been a shift because of the scanning protocol. And some scanning parameters in DTI, such as the voxel size, do influence some of the measures, such as FA. In fact, FA will be smaller if you're at a site with larger voxels. So in this review paper by Johanna Bayer and others, a number of retrospective data harmonization techniques are reviewed. One of them at the top, ComBat, assumes that the effect of age is linear. And so everything is sort of forced to be linear in the end using various site corrections. Or a little bit more realistically, ComBat-GAM, GAM stands for Generative Additive Models, Generalized Additive Models, assumes that the age effect is nonlinear. It doesn't have to be quadratic. It can be a fractional polynomial or very complex model. But after fitting an overall model to the data, site adjustments are also fitted that adjust the mean and the variance of the residuals so that all of the data can be referred to a common reference. So if you prefer algebra, this equation describes how ComBat-based site correction proceeds. The measures are on the left, y sub ijv, are a measure you've derived, such as cortical thickness, or it could be a microstructural measure from a certain site I, participant J, and region of interest V. That is statistically modeled as being influenced by covariates. It might be age and sex. The covariate effects there in X transpose IJ beta V model how age and sex that are in the data matrix X are influencing the measure Y. The estimated coefficients beta V loadings on, for example, age and sex. But notably, you'll see two terms that don't normally come up in single site modeling, the site effects. So the gamma IV at each site I shifts the mean values by a certain adjustment gamma, which is calculated during ComBat. And then the delta IV is a multiplicative term, which adjusts the spread or the variance of the data at the site. And so all at the same time, you can imagine estimating a statistical model of the data where the parameters you care about are in beta, the site dependent shift is in gamma, and the site dependent scale is in delta. And essentially, you're fitting the covariates such as age and sex and looking at the residuals, and then you're finding the shift and scale parameters that match the residual distribution to a reference. The reference can either be based on the data in the study, or it could be a high quality reference data set. And one small detail, if the batch size is small or the amount of data at a certain site is small, you can use a method called empirical Bayes to essentially shrink the mean and various correction factors towards values for the other brain measures at that site. That is not needed if you have a lot of data at each site. ComBat-GAM proceeds in roughly the same way. There's a more complex model is allowed, nonlinear model there in the middle, but the principle's the same. You still correct the individual site data using a term that adjusts its overall value, the mean correction, and a term delta that corrects the variance. And this allows a proper assembly of lifespan charts for brain measures. And so if each cohort spans just part of the age range, you'll see that this can align the data much better. And there are various extensions of this. You can correct the covariance of your data. You can think of these as normalizing transforms that map a site's data to corrected data. So if you do a lot of deep learning, you'll be familiar with batch normalization. You normalize the offsets in each data set. And just to wrap up, if you had data from a new site and you don't know what needs correcting, there's actually a formal statistical test that will tell you which moments of the residual distribution to correct, whether it should just be the mean, which is similar to random effects meta-analysis, or also the variance, skewness, or kurtosis of the data. And you would only fix the moments that deviate between your sample and the reference. And you can also use a standard reference batch. So when you do this, you'll find that using ComBat for data harmonization across sites makes it easier to detect effects of disease. So this toolbox by Joaquim Radoa in Barcelona has some easy-to-use functions that will correct for site and protocol effects in your multi-site analyses. And he's demonstrated that this provides increased power to detect disease effects. So just as you saw how the tensor distribution function eliminates sources of error in your data analysis from fitting tensors, this eliminates sources of error, or at least variance, that come from scanning people at different sites. And you could think of this as a sort of internal correction that can take place before you proceed with your group analysis of disease. So let's apply these to DTI. So what if we wanted to say, is this individual person's diffusion MRI scan abnormal compared to healthy people of the same age and sex? So there it is on the right. And then more sophisticatedly, what about their tractogram and their fiber bundle metrics? I mean, what about those metrics there? Are they statistically different than healthy people of the same age and sex? So one way to do this is using centile charts, and a closely related method is normative modeling. So ENIGMA's lifespan working group developed CentileBrain. It's a web page and software that shows you percentile charts for brain metrics from a little over 37,000 people from 35 countries. And in this case, it's the volume of the right thalamus, which increases till you're around 20 and then declines gently till you're about 60 and then more precipitously declines after that. And the colored curves are different percentiles of the healthy distribution, the healthy population that's been used to make these. So this is the raw data. It does follow the pattern. I mean, there's a little bit of a glitch where data from some sites is a little bit different. But this is the volume of the left hippocampus in a little over 10,000 people. As you can see, there's a gentle decline with age with volume. The different colors are data that comes from different sites and studies. So the ENIGMA Lifespan Group has used this to develop normative reference curves for in this case, volumes of different subcortical structures across the lifespan. The most recent paper on this was led by Ruiyang He at University of British Columbia in Lancet Digital Health. You can look at these and speculate about the causes of the different shapes of the curves. And you can also use the reference population to create z-scores that describe how much a particular individual or a group deviated from the expected reference. So just to say exactly how this is done, after site correction using ComBat-based harmonization, which is one approach to doing this, you can express the deviation of an individual from the normative curves as a standardized deviate, which is normalized by the root mean square error, which also depends on the age, and give each individual a deviation score or a z-score that describes their normative deviation. Julio Villalon at USC has computed these reference curves for DTI. He's made a normative model of DTI and brain microstructure in a little over 50,000 subjects. So the age range of the cohorts included is shown there on the top left. In fact, let's look at them. This model includes DTI data from a vast array of public data sets. On the top left, you'll see that the data sets don't all span the entirety of life. In fact, if you look at it carefully, a lot of cohorts might focus on adolescents or adults or old age. And the methods that we've been talking about, ComBat-GAM, for example, enable the integration of data from different parts of the lifespan to calculate a coherent trajectory. So let's look at the one in the middle. This is the average FA, fractional isotropy, of the white matter. It's a little bit of an odd measure to average that up across different parts of the brain. But if you do, you'll see that this measure rises in childhood and adolescence, gently declines into midlife, and then more precipitously declines in old age. And then in section C there, you'll see that there is actually a reference curve for each site. So why is that? Well, the reason is that the mean value of FA at each site might depend on the scanning protocol. And so using ComBat, for example, you can adjust a site-specific correction of the mean, and then either align that site's data with a reference and z-score it, or you can align the reference with your site's data. All of this would be calculated based on having a sample of healthy controls at your site, and then the distribution alignment will proceed using ComBat, which will align the mean and the variance of, or even higher order moments of the residuals. So this is very, very useful. So you can see how to construct these normative curves. Again, there's a separate set of curves for each region of the brain because the white matter maturational and decline profiles differ a little bit by region. But you can compile these and use them as a reference for incoming data. So what is seen biologically is that early in life, perhaps around age 26, the FA reaches its highest value throughout life. This is for the average FA. It may differ for particular regions of interest. But the RD, MD, and AD, the diffusivity measures, tend to reach their peak value. In fact, it's a lower point because those values have the opposite direction of change. And they tend to reach their peak value in the 40s and even 50s. So there's a sort of interesting difference in the maturation for each of these. So you can use these normative models. I mean, you could take the measures derived from your cohort, correct for batch effects in the way that we've described, and derive z-scores that quantify the degree of variation. You'll notice sort of an interesting observation here. So PPMI is a cohort of people with mild Parkinson's disease. And as we mentioned earlier, in Parkinson's disease, at least at the earliest stages, there's a rather odd effect where FA is higher than normal and MD is lower than normal. And so you'll see in Julio's example here, when the patients are plotted, the Parkinson's patients actually have an MD that is lower than normal, which is a disease effect. But then as the disease progresses, you'll see those dots sort of march through the center line and then eventually become higher because MD is elevated later in the disease. So you can do this for different diseases, different metrics, and observe the pattern of shifts that is going on. And there is some software to try this. So if you look up this paper by Alyssa Zhu in Neda Jahanshah's lab, she's developed a tool called eHarmonize, which is a tool to harmonize multi-site diffusion MRI metrics based on a reference data set of a little over 40,000 people. And you can see it takes the rather chaotic, unharmonized data on the left, and through a series of corrections, makes it conform to a more reasonable curve that can then be used for normative modeling. Okay, and this is Alyssa Zhu presenting the harmonization tools at one of the ENIGMA meetings last year. Okay, so in the final 15 minutes, we'll talk about more advanced methods for diffusion MRI. So these methods that we've described so far typically derive a summary value for each major region of interest of the brain, what if we wanted to map the microstructural changes throughout life or disease at a much finer scale? And so here are a few methods to go into this. So first of all, bundle analytics, which is a computational pipeline for diffusion imaging developed by Bram Chandio and her colleagues, Eleftherios Garifalidis and Jarek Haraslak at Indiana University, gives you an end-to-end solution for reconstructing your diffusion data, creating tractograms, making them into bundles, even cleaning up the bundles and turning them into tract profiles that you can then compare across subjects and across groups. And we'll tell you a little bit about the harmonization and statistical methods that are used there. A similar workflow also being piloted in ENIGMA is by Iyad Baghari in the Jahanshad lab, called medial tract analysis, where rather than looking at the whole set of streamlines, the medial curve is extracted and various measures of tract shape and geometry, as well as microstructure, can be analyzed on this medial curve. And then finally, Wendy Feng has been developing tools for normative tractometry. So much as we showed you for each region of interest, a way of flagging the anomalies in particular patients, you can even do this at a very, very fine scale level along tracts. And I'll tell you a little bit about how that is done. So first of all, bundle analytics, or BUAN, B-U-A-N, is a toolkit developed by Bram Chandio during her PhD work at the University of Indiana with Jarek Haraslak and Eleftherios Garifalidis. It takes a bundle representing a neural pathway in the brain, chops it up into sectors, and then if a disease is affecting some of these more than others, you have the ability to do a long tract statistics. In other words, rather than fitting a single statistical model for the whole thing, you could look at a certain region, in fact, all of the regions simultaneously, and plot at each point along the tract a statistic that describes to what degree aging affects the tract, or a particular disease, or a particular medication. And this is part of the DiPy toolkit and is available for anybody to use. And if you're at the DiPy workshop, there's a extremely in-depth coverage of not only tractography, but also these bundle analytic methods that Dr. Chandio will describe in considerable detail with hands-on tutorials. Just to talk about some of the steps that are involved, if you've collected diffusion MRI data, it can be reconstructed either with the diffusion tensor model or constrained spherical deconvolution, another method. In the end, each of these mathematical models of diffusion will help in estimating the neural pathways or the streamlines, the individual 3D curves that represent, for the most part, axons in the brain. This large mass of streamlines is then partitioned into tracts or bundles using anatomical segmentation methods. The bundles can then be cleaned. In other words, the outlier streamlines can be removed. And then this goes into the along tract analysis of microstructural and even geometric properties using these tract profiles. We'll talk a little bit about how the along tract harmonization and statistics proceed in the following slides. So these techniques give you a very refined anatomical parcellation of the brain in terms of the major neural pathways that you might be interested in, for example, the corticospinal tract, the arcuate fasciculus. I mean, many of these regions are preferentially implicated in some of the disorders, and you have now an ability to extract and model them. So some of the earlier tests in ENIGMA have been seeing whether these methods can be run multi-site. So for example, in bipolar disorder, if you run bundle analytics, for example, in different cohorts, and you fit, in this case, the effect of bipolar disorder to see where microstructural measures are linked with bipolar disorder. If you combine data from multiple sites, and I realize I haven't said how to do it yet, we'll go into that in a minute. It's encouraging that the more data that's added, or the more sites whose data are added, the p-values get lower, the effects get stronger, and the QQ plots in the middle are histograms of p-values that show that as data from more sites is merged in, the effects get more reliable. Now that's quite important. It could be that multi-protocol data integration just led to effects canceling out, but this is very important and encouraging work. Those of you who are familiar with bipolar disorder will see that the cingulin bundle, which is part of the limbic system, shows strong effects for FA reductions. And this could be a bridge to a finer scale mapping of the frontal limbic microstructure in bipolar disorder. Notice that there are also effects in the cerebellum. So, I mean, interesting new areas where differences are being found. So again, effects strengthen as you add data from different sites. I won't go into detail as to what the plots at the bottom show. Essentially, as you look at the bundle on the top left, if you go along its length and test for any evidence of statistical differences that are related to disease, the evidence varies at different parts of the structure, and you can make, as you can see below it, a sort of map of the p-value or degree of statistical evidence at each point. And certain methods for multiple comparisons correction, such as FDR correction, are then used to decide whether or not these differences are abnormal. But what you can see essentially going on here is that as data are added from more sites, the evidence for effects is increasing. How do you apply ComBat or other data harmonization methods to data along tracts? Now, you'll see in the workflow for BUAN that's shown on the bottom, there's a step in the middle, harmonization, where if you're operating with data from multiple sites or multiple scanners, you could introduce a harmonization step. Now, what does this actually mean? The data at a certain sector or region of the tracts is inputted into this approach. And you're allowing, just as we did before, the estimation of a correction at each region of the tract for the effects of site or protocol. And that's a correction of the mean and variance of the residuals after fitting a general statistical model of age and sex. And you can see that in the model right there, these gamma and delta parameters can be allowed to vary spatially. In fact, you could look at whether or not the correction for site depends on where you are in the brain. It's logical that the correction to FA for protocol effects of voxel size, for example, may depend on the number of distinct fiber populations with a voxel. They may not need correcting if there's only one fiber population, but if there are more, there may be more loss of signal in poorer protocols. And so you can imagine the gamma and delta could be estimated empirically by aggregating data from regions that are either within the same tract or have similar orientation density profiles. So this is an area of very active research. If you'd like to see how it's done, there's a preprint there at the bottom. These methods are beginning to be tried in different disorders. In the ENIGMA Autism Working Group, Gowan Kim and Kathy Lawrence have been trying this. The results on the top right come from standard tract-based spatial statistics. They'll give you a map of where the differences are in autism versus typically developing controls. But then look at the difference in the data on the bottom left. You'll see in the left arcuate fasciculus, there's a profile of spatially varying abnormalities in FA. And obviously, if you're interested in the circuitry and the regional anatomical localization, the refinement that you see is well worth running this workflow. In Parkinson's disease, Conor Owens-Walton and colleagues have been applying BUAN to do bundle analytics in 6 cohorts of patients with Parkinson's disease and age-matched healthy controls. And they're finding that in some of the pathways, for example, the spinothalamic tract, there are regionally varying profiles of microstructural anomalies. And obviously, given the very puzzling effects in early PD, there's some interest in sorting out whether they're localized or general, or what the different sources of those stage-dependent changes might be. Final topic, last 5 minutes, pertains to the use of generative AI for anomaly detection in tractometry. So those of you that use tractometry methods know that often spurious pathways or spurious streamlines are reconstructed, which is natural. The imaging data is not perfect. And the tractometry method will identify connections or pathways that are honestly not true. And it'd be very good if there was a statistical QC method that would flag those as outliers and eliminate them. And one method for doing this uses deep learning, a particular type of deep learning called generative AI, where you learn the statistical distribution of normal variation, and then you use it to flag whether or not there has been an anomaly. And one approach to doing this is a so-called variational autoencoder, which, as you see on the bottom, feeds in the input features, which in fact is the huge list of 3D points on a streamline, or in some approaches on the whole bundle. And then it learns a mathematical function that maps that vast list of coordinates to a very small number of parameters, which is known as latent space. So those of you familiar with principal component analysis will know that if you have a lot of variables and you want to do dimension reduction to lead to a much more limited set of variables, you can run principal component analysis and take a high dimensional input and convert it into 1 or 2 or 3 variables that you're then going to use to express the variation in the data. In much the same way, a variational autoencoder is a deep learning network that applies dimension reduction, but it allows the use of nonlinear functions, such as neurons in a neural net, to do the reduction. And you train it in a very unusual way, where you give it the input and have it reconstruct the input again, but you only allow it to use the very limited set of latent space parameters in the middle. And when you tune the parameters of this mapping, you use the degree to which it has faithfully reconstructed the output to decide what the parameters are. And in a sense, you're then learning an encoding of the data using a smaller number of parameters. That's why it's called a variational autoencoder. So when you look in the textbooks, this is Michel Thibault de Chotten's diagrams of neural pathways. They all look beautiful. But you'll know that when you run tractography, the extracted bundles contain spurious streamlines, outliers. So 3D curves that are obviously not part of the anatomical pathway. And there's some of them that are shown right here. So one way to eliminate spurious streamlines was developed by Bramschandio, and it's called FiberNeat. So it takes all of the 3D streamlines in the brain and observes that some of them are outliers, are spurious. And so a distance matrix is created on the streamlines. So for every pair of streamlines, you compute a distance. And there are distance measures for streamlines that have been used for many years. One of them, a minimum flip distance, was introduced by Eleftherios Garifalidis long ago. And so for every pair of streamlines, you can make a matrix of how far apart they are from each other according to this distance. And then that distance matrix is used to create an embedding of these streamlines into a much lower dimensional space. So methods for dimension reduction, nonlinear dimension reduction, include t-SNE or uniform manifold approximation, UMAP. And so every streamline 3D curve in A becomes a dot in B. And this embedding is constrained so that the distances between features in the original space, or relative distances, are preserved to the maximum degree in the embedding space. In other words, points that are close together in the embedding space are ones that the distance in the ambient space, A, are close. So what you're trying to do is map these streamlines to individual points in the embedding space in such a way that things that are close to them in the ambient space also appear close to them in the embedding space. And of all the methods to do this, t-SNE and UMAP are the oldest. Variational autoencoders are a little newer. And once you've done it, you can use a clustering method. A popular one is DBScan for clustering these and for outlier removal. So it turns out that the spurious streamlines all sit in that little patch at the top left of panel B. And after clustering, you can get rid of them. And this is tremendously useful. You can take a untidy bundle, like the one on the left there, run it through this method called FiberNeat, and eliminate these spurious streamlines. And There are other methods to do this. Jonathan Heitz has a presentation at the DiPy workshop where he discusses FINTER, which is a related method for cleaning fiber bundles, also based on deep learning. Now, you could say, well, if deep learning is good at finding outliers, why don't we use it to find effects of disease? And you can. So Wendy Feng, who's a graduate student in our lab here at USC, developed a deep normative tractometry method to identify white matter micro and macrostructural abnormalities in disease, here applied to Alzheimer's disease. And she uses essentially the same method, a variational autoencoder, or a variant of it called a convolutional variational autoencoder, to encode the normal range of variation in number one, the shape or geometry of these bundles, and number two, the microstructural profiles, the profiles of FA and MD and the microstructural metrics. And then using a variational autoencoder to decide whether or not a particular individual's streamlines are normal or within the normal range, the deviations are then plotted point-wise along the tracks. And if you're interested, there's a preprint on deep normative tractometry at the link there at the bottom. So just to sort of recap how these work once again, this is work from the UCLA group led by Shantanu Joshi, which uses a vector quantized variational autoencoder to encode streamline geometry. And there are a number of ways to do this using deep learning, but the fundamental principle is that you compress the data so that it can be represented using a much smaller set of variables, and they're often called latent space variables. This produces a compact compression of the data from which you can reconstruct the data using a more economical set of parameters. The nice thing about variational autoencoders that is not present with TSNE or UMAP, which are related dimension reduction methods, is that it also produces a probability density for the streamlines. So a variational autoencoder takes data and maps it onto a multidimensional isotropic Gaussian in the latent space. And for every input, whether it be a streamline or a bundle, it'll ascribe a Gaussian deviation score. And so just by construction, you will get a normative distribution on these streamlines, which is then very useful for detecting abnormalities in the disease. So just to recap, deep normative tractometry, which was developed by Wendy Feng, jointly models white matter microstructure and the whole brain macrostructure, the geometry of the bundles, using variational autoencoders. Variational autoencoders are a nonlinear extension of other simple dimension reduction methods, such as principal components analysis, PCA. When you have a lot of data, you can use nonlinear methods, see if they're any better, and VAEs are one such method. These can learn the normal variations in tracts. So if you feed a lot of tracts and streamlines from healthy controls, you can use them for normative modeling. You could even condition an autoencoder on age and sex by inputting those variables as well, and saying, please learn the normal distribution of fiber variation for a person given this age and sex, and then detect anomalies and produce an along-track mapping of those anomalies for differences in microstructure and macrostructure. And so if you look on the bottom right there, this is one streamline in a bundle from an individual patient. And the z-scores come from comparing that patient with a normative reference that's been derived using deep normative tractometry. And this is incorporated in DiPy. It's compatible with RICO bundles and BUAN outputs, and you're more than welcome to use it. So this is just a quick comparison of deep normative tractometry and BUAN bundle analytics for identifying differences in these tracts. Why are they different? They're a little bit different. And so if there is a local anomaly in these tracts, bundle analytics proceeds in a mass univariate way and will compare data from that point on the tract with all the other people in the study, but without regard for the rest of the bundle. Deep normative tractometry takes the entire bundle as an input and uses a variational autoencoder to encode what are the allowed types of variation, either in microstructure or geometry. And if the overall configuration of the bundle is anomalous, it will notice that. And then just so we can see it, it'll plot the mean absolute deviation along the tracks and help you see where the differences are. Now, this is not an entirely new idea. We weren't the first to do it. Maxime Chamberlain, for example, developed a deep learning approach that detects anomalies in tracks that's there at the link if you want to take a look at his work as well. So just in summary, in this review of international neuroscientific projects, we focused on diffusion MRI. Certainly diffusion MRI powerfully detects effects of disease on the brain. Not every analysis requires data from multiple sites, so it's particularly useful to combine multi-site data if you're looking at a question that requires high statistical power or a subtle effect like a psychiatric condition. You may not need multi-site analysis if you're studying, for example, Alzheimer's disease or stroke, where the effects are very large. Single-site low-N analyses can often lack power, and they can often have unknown generalizability. So many factors, as we saw, influence the disease effects. And so in multi-site analyses, you can model some of these, and you probably get a better idea of whether or not the experiment will replicate. Now, just from a technical standpoint, harmonization tools and normative models can help you to integrate and compare data from many sites. Particularly with DTI and diffusion MRI, the acquisition protocol of the scanner does influence these. And there are many, many methods to adjust for this, either to align data from a particular scanner to a reference. They often use data from a sample of healthy controls that have been scanned on the scanner with that protocol. And that gives you a principled way to align the data distribution from a particular site with data from other sites using a large range of mathematical techniques. Some of them just shift the mean of the residuals, others shift all the higher order moments as well. Now, these site correction methods are now being extended to tractometry. We looked at Brown-Chandio's method for harmonized tractometry, which uses ComBat at different sectors of the tract. But there are also methods using generative AI that, rather than operating pointwise, take the whole distribution of the fiber geometry, embed it into a latent space, and then look at the conditional distribution of those latent measures conditioned on age and sex to model the distribution of fiber geometry and microstructure within the brain and flag outliers. So just a word of thanks. Much of the work presented today was done by Dr. Brown-Chandio, Julio Villon, Wendy Feng, Leila Nabilsi, Connor Owens-Walton, Gaon Kim. Members of the Jahanshad Lab contributed a great deal to the methods that were shown today. And obviously, we'd like to acknowledge the NIH and all the other agencies that fund ENIGMA. There's a picture on the bottom there of a gathering of ENIGMA members, and then for those that can never all meet in person, this group pictures in the middle. Our group at IGC in Marina del Rey is pictured there at the top. And a big thank you, of course, to Eleftherios Garifalidis at Indiana University and his statistical colleague, Jarek Hauslak, who in fact mentored the development of many of the tools that were shown for bundle analytics developed by Brown-Chandio that was shown today. So once again, thank you for your attention. If you're ever in Los Angeles, you're more than welcome to stop by, and we'll talk about anything you're interested related to the topics of today. So again, thank you very much for your attention. Bye-bye for now.